Okay, our next podcast is going to go into gene function and looking at the process of protein synthesis. So we can start by defining what a gene is. It's a sequence of the DNA molecule, so it's a segment on the DNA. And in that, it codes for a particular protein, which is going to be involved in the production of a particular trait or in a metabolic function or whatever. But there's two broad steps that are involved with the production of protein. First, we start with our DNA, but then we transcribe that DNA into our RNA. So RNA is still a nucleic acid, but it has a different sugar than DNA. It has just ribose instead of deoxyribose. So the first process is to transcribe the information. So that code or that sequence that is that DNA is first transcribed into RNA. Now, when you transcribe something, you stay in the same language. So your friend misses class and you have to take notes for him, you take notes, and he transcribes those notes. He rewrites them in his own notebook in his own language. So that's transcription. And this is staying in the same language. It's still staying in the genetic language or the nucleic acid language. So we transcribe. That's the first broad step in making a protein. The next step, let's see, let's go. Yeah, okay. So the next step, we do an RNA processing where we make we complete making our RNA, and but that's still the latter part of transcription, and then we go into translation. So transcription and then translation. Now, if that friend of yours is Chinese and they take those notes and they have to rewrite them in Chinese, that's translating it into a different language. And so this broad process of protein synthesis is going to translate the genetic information that is now on the RNA, translate it into a protein. It's going to build up a bunch of amino acids to make a protein. And so that's transcription. So that is the broad steps of protein synthesis. Now, we can look closer at what's happening with that DNA. So here's our gene. We look at this one little gene of interest right here on our long strand of DNA. And here's our sequence of ATCs and Gs that make up the DNA of this individual. And then we make our RNA first. We transcribe this information into still genetic information, but in a different molecule. When we transcribe this, we complementary base pair the nucleic acids, the bases that are on the nucleic acids of our template. So here is our template. So we transcribed C's always pair up with G's. C pairs up with G's. Now, A's, they pair up with T's in DNA, but A's pair up with U's in RNA. So that's different. RNA does not have thymine. It has uracil in place of thymine. So we match these up along our strand of DNA, and that makes our RNA. So that's the first broad step. Then we translate that information. Now, when we translate it, the genetic information is based in threes. So we have 3 letters that make up a codon, just like we have 26 letters in the alphabet. We can make up a bunch of different words. They can be a lot of different links. In genetics, their words are always 3 letters. We call them codons, and we only have 4 letters to work with. And so a codon is made up of 3 of those 4 letters. So the first codon here is UGG, the next codon is UUU, and then GGC. Those are the codons. Each codon will code for 1 amino acid. So in the translation process that takes place in the ribosome outside of the nucleus, each codon will code for 1 amino acid, and then they build upon each other to make a string of these amino acids, which is going to ultimately make our protein. So those are the broad steps of protein synthesis. Now, there are a certain number of amino acids, and we know those, and we know what each one is coded from the RNA. And so this is our template here of all of the possible codons with those. So this would be all the possible words in genetics you could make in an RNA molecule. So this is all the possible outcomes of our 3-letter words, choosing those 4 letters. And this has which particular amino acid is coded for those codons. So these 4 codons are always going to code for that amino acid. And so these are all the amino acids that are part of the genetic code. Now we can look at where this occurs, and when this is occurring, when we are making this RNA, this first RNA strand, and we call this RNA strand the mRNA because it is a messenger RNA. There's another RNA that plays a little bit different role. It's called a tRNA, or transfer RNA. We'll look at it in a minute. But when we are copying the DNA to make our RNA, this is what the process looks like. We have an enzyme that comes and attaches to our DNA. It starts at a starting point. There's always a starting point that begins the process. And so that's a particular codon on the RNA that begins that process. And so this occurs. We have our let's see, we have our polymerase is the enzyme here. The gray enzyme here is our polymerase that's going to kind of help open up the DNA as it moves through. The first broad step of the process is called initiation, and so that's when everything begins here, and we have our beginning. All right, so that little spot is the beginning of our RNA right there. And so what happens is the enzyme moves down. As it moves down, it's reading the DNA and building an RNA. So coming into one side of the enzyme is the RNA nucleotides that are coming in, and coming out of the other side of the enzyme is the combined nucleotides, the actual RNA strand. This shows a blown up view here toward the end. So we have initiations, the beginning. Elongation is the building of it as it moves along, and then termination is the end once it separates. So once this is built here and it's coming out the end, we have our newly made RNA, and then once it gets to a stop point, there's also a codon for stop. Once it reads that, then it stops, and this is complete now, and it separates. Now the RNA is made, and it has the message. Basically, it just transcribed that message from the DNA. Now it's on this red strip, the RNA, and it can go to do what it is what it's coded to do. Okay, and then the other broad step of protein synthesis is to take this information from this RNA and make a protein. So this now leaves the nucleus. It goes to a ribosome that is outside of the nucleus. And here it is right here. It goes through a fold in the ribosome. And once it starts moving through in this direction, we have some tRNAs that come in. Now, tRNAs are transfer RNAs. These are molecules that have an anticodon on one side and they have an amino acid on the other side. And they have this specific amino acid that is coded for by the codon that is on the RNA. So this comes in, it matches up with complementary base pairing with the codon that is here, and it brings in one amino acid, and it slides down, and the amino acid hooks to the chain, and the next three are red. And another tRNA comes in and hooks with those, slides over one, and the next three are red. It's kind of like a typewriter ribbon going through a typewriter. It moves through. As it moves through, it reads these codons, and a tRNA is going to bring in an amino acid till we build a long chain of these amino acids. And we use the same words for initiations when it all begins, elongation when it moves through, and then termination, it separates. When termination occurs, we now have our protein is now produced, and the process, the end of protein synthesis has happened. Okay, we can see that here. This is initiation, the beginning here. We can see the first tRNA is coming in, and another one, the red arrow here shows where the next one will come in, and then that will slide down, and we have the next one will hook to there, and it'll continue to slide over, and we'll continue to build upon that until we get a long chain of these amino acids to actually build that, and now we have our protein made. This is a slide here at the end of the chapter that just kind of summarizes all of that. The transcription is taking place here in the nucleus of the cell, and then the RNA is made, and it leaves the nucleus, goes out to the ribosome, and here's where translation takes place. All right, that's where we end there. Still have a little bit more to talk about with genetics, with gene expression, and then also genetic engineering. So those will be in some future podcasts.